In this chapter, we will look at the basic principles of heredity, focusing in on Mendel's work. So, Gregor Mendel was an Augustinian monk. He lived in the 1800s, and he is considered the father of genetics. And so, why was his work so important? So, he figured out the very basics of how heredity works. And, sadly for him, his work was very largely unappreciated during his lifetime by other scientists. It was not until about the early 1900s that his work was rediscovered by the scientific community, and he got the credit due to him at that point. So he was so successful for a couple of reasons. First of all, he used an experimental model that worked out pretty well for him. He used the garden pea, and the garden pea, first of all, it gives a whole lot of progeny for every plant that you cross, and you can get the results in one growing season. Next, he used a mathematical approach. So he actually counted the number of progeny and looked at the actual ratios between different types of progeny. And finally, he studied very easily distinguished characteristics. So these are things that it's just kind of a yes or no. Does it have purple flowers or does it have white flowers? There wasn't a continuum between these. It was just plants had either purple flowers or they had white flowers. Mendel had available to him a whole bunch of what we call true breeding lines, also called pure lines. These ones had been used in agriculture for a long time, and they had different characteristics. So you could get a line of peas that made peas that with round seeds, or you could get one that had wrinkled seeds. Likewise, you could get one that had yellow or one that had green. All of these different traits are the traits that he looked at, and the phenotype, okay, you've probably heard this term before, it's the form that is taken by a particular characteristic or a trait. So Mendel did two kinds of crosses. First of all, if you just leave a pea plant to do its own thing, it will actually do a self-cross, and you have a flower right here, and the pollen from the flower will directly fertilize the stigma on that same flower. Cross-pollination happens if you actually remove these anthers before they mature. You can cut them off as soon as that flower opens, and then use the pollen from another plant to fertilize this flower. So Mendel was interested in what would happen if he crossed two different pea plants together that had different characteristics. So he crossed one that was round to another plant that gave wrinkled seeds, and he looked at the results. So here's what he found. So first of all, I'd like to say a few things about how we talk about different generations in a genetic cross. The P generation is the parental generation. The F stands for filial generation. Okay, so that's the immediate progeny of the parental generation. And then the F2 generation, filial 2 generation, is the progeny of the F1 generation. So he crossed in the parental generation, he crossed true breeding lines, one from a plant that had round seeds, crossed it to a true breeding plant that had wrinkled seeds. What he found is that one of the traits disappeared in the F1 generation. So in the F1, he saw all of the progeny had round seeds, and then he allowed these to self-fertilize, so he crossed them among themselves. And he found in the F2 generation that the wrinkled phenotype showed up again. So not only did it show up again, but it always showed up in a particular ratio. So there were 3/4 of the round phenotype, and 1/4 of the F2 generation was wrinkled. So based on these results, Mendel formed his first hypothesis about how heredity occurs, and this is called Mendel's first law, or the principle of segregation. There are a number of aspects to this law. First of all, Mendel thought that genes or genetic factors come in different forms, and we're going to call these different forms alleles. So genes for a trait, Mendel proposed, they come in pairs. And so every individual organism has two of each gene. And when those genes are together in an individual, they do not mix together, but rather they stay separate from each other. And during gamete formation, that would be during meiosis, those two alleles will separate from each other, and half of the gametes will contain one allele and half of the gametes will contain the other allele. Mendel also proposed that when you have two different alleles that are both present in an individual, one of those alleles is going to determine the phenotype, and that allele is going to be the dominant allele, and the other allele is going to be present, but it is not going to affect the phenotype, and that is a recessive allele. So one thing that we will do quite a bit of in class is writing out the genetic notation of a cross and predicting what the progeny will be from a cross. So when we write out a genetic notation, remember that each individual always has two alleles of each gene, and the term for this is called the genotype. Dominant alleles are going to be capitalized, recessive alleles are going to be lowercase, and if you have an individual with two of the same allele, we're going to call those our homozygotes. If you have an individual with two different alleles, we call them a heterozygote. When we look at this cross, true breeding round seeds crossed to true breeding plant with wrinkled seeds, we know that wrinkled is the recessive trait because it disappears in the F1 generation. And so we are going to write the genotype. This plant has two of the R gene, and it is true breeding, so it's going to be homozygous. So big R times little r. How are we going to predict what we get in the next generation? So we commonly use Punnett squares in genetics to predict the progeny of a cross. And even though most people have learned how to do this all the way from middle school, I find that there are a lot of people that continue to be confused about how to use Punnett squares properly. Punnett squares can help you predict progeny of a genetic cross and also their relative frequency. Now, Punnett squares can differ in their size depending on how many genes you are looking at in that cross, and also what the genotype is of the individuals in that cross. So in a Punnett square, you would put all the possible gamete genotypes from one parent listed across the top, and all the possible gamete genotypes from the other parent listed across the side. So in this particular case of this parental cross, the possible gametes are as follows. All of the gametes from this big R individual are going to be big R. There's only one possibility. All of the gametes from the wrinkled individual are going to be little r. And so the way you would fill out this Punnett square is very, very simple. In this case, you would just put big R for the gamete genotype of the round parent and little r for the gamete of the wrinkled parent on the side. And then you just combine them together to make the genotype of the progeny. So in this case, there's only one type of progeny. And so all of the progeny in the F1 generation are going to be big R, little r, or heterozygous. In the next generation, we self-fertilize, so the heterozygote is crossed to another heterozygote. If you look at each of these, each individual here has two possible gametes. Here we've got two different alleles, and so those alleles, according to Mendel's law, separate from each other during gamete formation, and half of the gametes have one allele, half of the gametes have the other allele. So half are going to be big R, half are going to be little r for one parent, and the same thing for the other parent since we are selfing. So this will make a different kind of Punnett square, and this is going to be a 2 by 2 Punnett square, or 4 possibilities. You join these two together and you get 4 possibilities. 3 of them are going to be the round phenotype, and 1 of them is going to be the wrinkled phenotype. Now, I'd just like to point out that Mendel said that genes come in pairs, and those gene pairs separate or segregate from each other during gamete formation. We've already talked about mitosis and meiosis, and we've talked about the fact that chromosomes also come in pairs, and these pairs are called homologous chromosomes. One comes from your mother, one comes from your father. And in fact, those gene pairs are found on the homologous chromosomes. And so if you're a heterozygote for the A gene, one of the A alleles will be on one homologous chromosome, one of them will be on the other homologous chromosome. And it is the movement of homologous chromosomes during meiosis 1, when the homologous chromosomes separate from each other, that ensures Mendel's law and ensures that the two alleles segregate from each other during gamete formation. I'd just like to take this opportunity to point out one other important term we will use. So a genetic locus is another term for a gene, and if you're talking about several genes, the plural is genetic loci. The important point about this term is that it highlights the fact that genes are actually found at a particular location on a chromosome. Mendel also did a different kind of cross where he looked at crossing two individuals that differed in two different traits. So not just round versus wrinkled, but also including a different trait such as yellow versus green. The results of these experiments demonstrated what came to be known as Mendel's second law, which is the principle of independent assortment. If you look at this cross, you'll notice it has a lot of similarities to the single gene crosses that Mendel did. So you start out with the parental generation of true breeding plants, and in the F1 generation, you only show the dominant phenotypes. So the recessive traits, the wrinkled and the green traits, disappear, but then in the F2 generation, when you cross those F1s among themselves, you find that the recessive traits show up again, and they show up at this distinctive ratio of 9 to 3 to 3 to 1, where 9 is the double dominant, the two dominant traits, and the two threes are one dominant, one recessive trait, and then the one class is the double recessive trait. If you look carefully, you'll notice that the ratio observed here in the F2 generation is actually two independent 3 to 1 ratios. So if you add up all of the round, and you compare that to wrinkled, you would get 12. So 9 plus 3 is 12, and 3 plus 1 is 4. So 12 to 4, or 3 to 1. Same thing with the other trait. So since this represents two individual 3 to 1 ratios, Mendel proposed that the gametes are actually segregating independently of each other. So if we then go into the genetic notation just briefly here, if we look at the parental generation, these are both true breeding individuals, and now we're looking at two genes. When you're looking at the gametes, it's important to note that each gamete has to have one of each gene. So you have to have an R gene and you have to have a Y gene in that gamete. Now if you look at this particular one, you note that there's only one kind of R allele that can come from this parent and one type of Y allele. So all the gametes are big R, big Y. All the gametes from the other parent are little r, little y, and you get in the F1 generation individuals that are all heterozygous for both genes. We also call this a dihybrid individual. Then you self-fertilize between the individuals in the F1 generation, so you cross a dihybrid to another dihybrid, and here we are now going to have more complex gamete genotypes. So since the R and the Y genes are independent of each other, if you have a big R can either go with big Y, or big R is equally likely to be with little y. Same thing with little r. If you have little r, it's equally likely to be with big Y or to be with little y. And so you have 4 different kinds of gametes from a dihybrid, and those are all equally likely. You cross one dihybrid to another dihybrid, so the gametes from the second parent are on the side here, and you would complete the Punnett square, and from that Punnett square you would get this 9 to 3 to 3 to 1 phenotypic ratio.